Welcome to our video lecture on neural development, option A. We're actually going to start with a little bit of a review of neurons and synapses, and then we'll get into all the amazing details regarding how neurons develop. Before we get going, let's take a look at our objectives for this lecture. So this is our essential understanding, that big picture idea that IB wants us to come away with at the end of this lecture. So modification of neurons. That differentiation piece that gets neurons to be neurons starts in the very earliest stages of embryogenesis and then continues to the final years of life, which is really amazing. So some of our details, we're going to review neuron structure. We're going to talk about nerve impulses, our resting potential, depolarization, repolarization, hyperpolarization. We're then going to switch over to embryogenesis, embryo structure, embryo development, and how neurons develop in those embryos, and that process is called neurulation. We're going to look at brain, spinal cord, and then vertebrae, and then at the very end we're going to talk about how neurons and axons get connected to each other, how some of them might get pruned or pulled away, and then brain plasticity, how we can kind of edit the things that our brain can do, those connections in our brain. So lots of words, many of which you should remember, but if not, feel free to pause the video, look these guys up, and then restart the video. But for now, here we go. All right, so let's review neuron structure super quick. So this right here, remember, is going to be the nucleus. The nucleus is going to be found in, we call this the soma, or the cell body of the nerve cell. These guys right here, the very ends of the cell body, these are dendrites. Dendrites are going to collect information from the previous nerve cell. This is known as the axon, which runs all the way down through here. These guys at the very end of the axon are known as axon terminals. Sometimes we spell it terminus. And if this nerve cell is talking to a muscle cell, then we can also call this the motor end plate. Along our axon, we have these chunks of cells. These cells are Schwann cells, and they make up the myelin sheath. Remember that the myelin sheath allows for nerve impulses to jump, 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 jump. That speeds up the rate of our nerve impulses. This is known as saltatory because in Espanol, saltar means to jump. And then here, these little divots between those Schwann cells, these are the nodes of Ranvier. And this is where that nerve impulse is going to jump. Good? Good. All right, so a couple different kinds of neurons, and we're just going to talk about a typical neuron. But there are sensory neurons. The job of these guys is to collect information from our sensory organs. So we would have an eyeball here, or maybe we'd have some skin. That's supposed to be a hand with some receptors in our fingers. And so that's going to pull information from our sensory organs toward the cell body to the axon terminals, which is going to then connect to an interneuron. Interneurons are in our spinal cords. These guys will also send signals up to our brains, and then they can also send signals onto our motor neuron. These are the nerve cells that tell muscle cells what to do. And this is probably what we are used to seeing most, where we've got the cell body, we've got those dendrites, we've got axon, myelin sheath, nodes of Ranvier, axon terminals. So not all nerve cells look like our motor neurons, but that's definitely the one that we like to look at in our pictures most often. How do we know how impulses pass? Well, let me tell you, this amazing woman right here, her name is Ida Henrietta Hyde. She invented this micro electrode. And so it is a machine with teeny, teeny, tiny little needles. And the needles can go into and out of the axon of a nerve cell, and we can actually record with this microelectrode the difference in charges due to ions inside and outside of the axon. So how we know that this is resting potential at about -70 millivolts, and this is depolarization and repolarization, hyperpolarization, and then back to resting potential, is all because of this amazing woman, Ida Henrietta Hyde. Let's talk about these different pieces. Remember that this is going to be resting potential was just about -70 millivolts. So if we're able to get past that threshold potential, then we're going to have this rising action. This, of course, is depolarization. This is depolarization. And this is when, remember, the sodium channels open. Sodium rushes into the cell. Here we're going to have our sodium channels close and potassium channels are going to open. Potassium inside the cell is going to leave, and that's going to cause the cell's charge to go back down. This is repolarization. Then we have a little divot here, hyperpolarization, sometimes also called the refractory period. This is when the cell is just a little bit extra negative before it gets back to resting potential. When we are in this extra negative little chunk of the graph, the cell cannot send another nerve impulse. We need to get back to resting potential before another impulse can go past. This whole thing is known as the action potential. And then we go back to resting potential. So here are some of those details one more time. So to get to resting potential, how do we even get to -70 millivolts? Well, we have these amazing sodium-potassium pumps. Sodium-potassium pump. Remember that pump implies energy is needed. ATP is going to be used, and this is going to be moving things against the concentration gradient. Energy is needed. We're going against the concentration gradient. The sodium-potassium pump is going to take 1, 2, 3 sodium ions and push them out of the cell. It's going to take 1, 2 potassiums and pull it into the cell. And this, of course, is going to use some ATP to make it happen. 3 sodiums out, 2 potassiums in, 3 sodiums out, 2 potassiums in, because we have more positively charged stuff out. The outside of the cell is positively charged compared to the inside of the cell, which means that the inside of the cell is going to be negative and just about a -70 millivolts in comparison to the outside. There are some other ions that are getting moved around, but the two most important ones are sodium goes out, potassium comes in, and that gets us to -70 millivolts. So then when the nerve cell is stimulated in that action potential, the action potential happens, that nerve impulse is passing along the cell. We're going to start with depolarization. Depolarization because we're going to flip the sign of the cell. So right now we're about -70 millivolts. We're going to get that guy up to maybe a +30 or even 40 millivolts. How that happens is we have these amazing sodium channels. They are voltage gated, which means that the channel will open or close depending on the stimulus, the charge that is being propagated along the cell. So we're going to open up sodium, and then all the sodium that we pushed out is going to get in. So all the sodium that we pushed out, 3 sodiums out, 2 potassiums in, all that sodium we pushed out is going to get in. And now we're going to have more positive things inside the cell because we allowed that sodium to come in through that channel. That's going to make the cell be more positive because it's got more positively charged things on the inside. And that's why the charge goes up to about +30 or maybe even 40 millivolts. Good. This is called depolarization because we go from negative to positive. And then what's going to happen is our sodium channel is going to close back up and potassium is going to open. So now we're going to go from that positive that we got back to negative, which is why we call it repolarization. So we're going to close the sodium channel. No more sodium gets in, but we're going to open the potassium channel. And all that potassium that we worked so hard to get into the cell, it's going to get out. which is going to cause the cell to lose positive things, which makes it become more negative, repolarization. And then we're going to have this little dippy-doo refractory period, because even though the sodium-potassium pump is working really hard to push things back in place, they're just not quite there yet. And so we have a little extra negative because there are so many positive things on the outside, extra negative, and we cannot stimulate that nerve cell for just a little bit of time until the sodium-potassium pump, 3 sodiums out, 2 potassiums in, 3 sodiums out, 2 potassiums in, gets us back to negative 70 millivolts, and then the thing can happen all over again. All right, we're going to put away our nerve cell review and talk about how do we even get nerve cells and how do we study nerve cells in terms of differentiation. So we tend to not use humans when we're looking at how nerve cells are growing and developing. Instead, we have some model organisms that we really like to use. And so developmental neuroscience, there are lots of ethics that prevent us from using human tissue to study our developing neurons. And so we use these guys instead. So this is Caenorhabditis elegans, also known as C. elegans. Usually I have to practice that a couple times, which is this guy. He is a roundworm, has just a few chromosomes, so it's really easy to study because there's just a couple chromosomes to look at. Drosophila melanogaster. These are fruit flies. Sorry, my phone is going off. These are fruit flies. They also have just a few chromosomes. They only have 4 chromosomes altogether. They also make babies and mature really fast and in great numbers. We can have super big sample sizes, crazy, crazy fast. Denio rario is zebrafish. These guys, what's super cool about them, their eggs, their embryos are almost transparent. So it's super easy to see their tissues developing just by looking through microscopes. We don't have to slice them open. We can just see them because they're so transparent. Xenopus is this guy, this frog, African cloud frog. They're super cool. We like them because their eggs are giant and they're super easy to manipulate. And then Mus musculus, these are mice. We like to use mice because they have lived with humans for thousands and thousands and thousands of years. And so we actually share a lot of diseases with them. So mice are good to use because the diseases that we humans have, often mice also have. All right. So let's talk about some development. So we're going to start with a zygote. Zygote is egg plus sperm. So that very first fertilized egg, and you can kind of see the 2 nuclei here. So one is the egg and one is the sperm, and they are about to merge into a single nucleus. So this is our zygote, just 1 cell big. And it's going to go through mitosis. Now we have 2 cells, mitosis again, 4 cells, and we're going to eventually have so many divisions that we have this ball, this solid ball of cells, and we call this the morula. After another day or 2, day 3, 4 in mice, day 5 in humans, we're going to have what we call a blastocyst. And what has happened is this solid ball of cells turns into a hollow ball of cells. And so all the cells that were filling up the ball migrate to the outside of the ball. So going from here to here, the inside of this guy, the blastocyst, looks like this. This the empty space in the middle is known as the blastocele. What's kind of cool is that the cells will start to divot into the blastocele. At this point, it's known as not a blastocyst, but a blastula, and then that divot is going to start to grow extra layers of cells, and now we have the gastrula. And this is where neuron differentiation really starts to happen. These cells that are near the divot, these are now going to be nerve cells. They clearly do not look like nerve cells yet, But because of the positioning on the gastrula, because of the chemical signals that are about to infiltrate these cells, these guys are destined to be neurons. So they've lost some of their stem cell potency. These guys at this point, we cannot turn them into bone marrow. We cannot turn them into digestive cells. These are bound to be neurons. So quick look at those 3 different layers of cells. Pretty cool. So all the cells in the endoderm, so these guys in the very middle of the gastrula, they're going to become the internal layers of cells. So digestive cells, thyroid, lung cells, things in the very, internal tracts of our bodies. Mesoderm, meso in the middle. This is going to be lots of muscle, some of our kidney cells, blood cells, lots of muscle and bone. And then ectoderm, this is what we're going to focus on most, because ectoderm actually turns into neurons and skin cells and some pigment cells. But most important for us in today's lecture, these ectoderm cells are going to differentiate into neurons. So again, this is just another look at another view of the slide that we just saw. So this is Xenopus. This is, again, that frog that we like to look at because they've got really big eggs. So blastula, and then this is going to divot. There's the little divot that we can see in the gastrula. And then what we're going to focus on is the formation of this neurula. So what's going to happen is this little divot is going to start to spread out. We're going to have some holes. We're going to have like a thicker ridge of cells just because of how the cells are moving. And this is going to eventually become spinal cord, vertebrae, and then brain. So we're going to focus especially on, again, how we get from here to here. So how do we get from there to there? That process is known as neurulation. Neurulation is differentiation of cells into nerve cells. It is the formation of the notochord. It is formation of the neural plate. It is formation of the neural tube. It is formation of nerve cells. So on the previous slide, I kind of showed you that divot, and they had the divot running this way. Well, now we're going to kind of cut, slice through that little divot is this line right here. So the most important starting piece is the notochord. Notochord is a little bit of a mesoderm that starts to form right after that gastrulation, that dip starts to happen. The notochord is going to send some signals, chemical signals out, which is going to cause the ectoderm's in blue here, it's going to cause the ectoderm to fold up, and then it's going to keep on folding, and eventually it's going to merge like this. And so again, and get rid of my scribbles so that you can see it one more time. We're going to have notochord here. It's going to send chemical signals out, which is going to cause this ectoderm, remember that our nerve cells come from ectoderm, it's going to roll up and around until we have a tube, and that tube is known as the neural tube. What's kind of cool, the neural tube is going to eventually become spinal cord and brain. That little hole in the middle here, that hole, that cavity still exists in our spinal cord. If you've ever had to get a spinal tap, this is where we sample some of the fluid that's in the spinal cord to see if there's an infection of the spinal cord. This is where they're pulling that fluid from. That spinal fluid is there. So notochord is mesoderm, neural tube is going to be ectoderm. This is going to turn into skin. And what's super cool, so this mesoderm stuff here, is known as is going to develop into what we call a somite. So these are going to be some somites on the side of the neural tube, which is going to become the spinal cord and brain. These somites, which is mesoderm, ectoderm, mesoderm, this mesoderm is actually going to develop into the vertebrae and tendons and ligaments and muscles that surround our spinal cord. So you've got like all the makings for a backbone, spinal cord, bones, tendons, muscles, all right there. So we're going to look at cross-section one more time. So dorsal, this means back, which makes sense because this is where we're going to find our spinal cord is on our back. And so neural tube is going to be here. And that little divot in the middle is where we're going to find that spinal fluid at some point. Here's notochord. Here we have those somites. The somites are going to become vertebrae, other backbone muscles and tendons and whatnot. Our kenteron, this is just like what's left over of the little cavity. It's eventually going to become basically the inside of our digestive tract. So this will be like the inside of esophagus, small intestine, stomach, whatnot. Endoderm, ectoderm, mesoderm. What we care about the most is this stuff right here, this stuff right here. We've got our neural tube, which is going to develop into spinal cord and brain. Somites are going to develop into vertebrae, those bones of our backbone, muscles and tendons that hold those bones in place. And then notochord is what's going to stimulate all of that differentiation to start. Good. So looking at these vertebrae real quick. So our somites that are going to differentiate into bone and muscle and ligament, some of that being our vertebrae, there is a disorder known as spina bifida. Spina bifida is a disorder in which the vertebrae do not fuse all the way. So what should happen, this is normal, what should happen is the vertebrae grow all the way around and we have our spinal cord right in here. If the vertebrae do not grow all the way around, then the spinal cord can protrude a little bit out of the backbone. And there are a couple different severities of spina bifida. So there's a little bit where there's just a little bit of a bone missing, and so you might have a little bit of bulging of the spinal cord, but it stays in the spinal cord area. It can get worse, and the fluid of the spinal cord, that cerebrospinal fluid, could actually grow into a little bubble that is noticeable from the back of the person. So you can actually see this bubble. The worst case of spina bifida is when there's not just fluid, but also nerve cells that are protruding through the back of the person. And of course, this is just skin. There's no bone here protecting the spinal cord. And so any kind of injury could allow for infection or damage. So this kind of spina bifida is very, very dangerous. And it seems to be folic acid. Folic acid is one of the vitamins that women who are planning to have babies or who are currently pregnant, folic acid is super important. It seems to help prevent spina bifida just a little bit. So spina bifida is a disorder where the somites do not develop into complete vertebrae, and the spinal cord can be exposed, and that can be a risk. All right, let's talk about, instead of spinal cord, let's talk about specific nerves. How do these nerve cells that are going to become the spinal cord, how do those nerve cells of the ectoderm turn into neurons? So differentiation is going to start to happen in the neural tube. Those nerve cells in the ectoderm are going to be nerve cells. They don't have any option. Mitosis happens and happens and happens and happens and happens, and we get billions and billions of neurons. So we're going to have that multipotent progenitor cell, which can turn into lots of different kinds of nerve cells, but only nerve cells at this point when we're talking about those neurodermal cells, those neuroectodermal cells. We have a couple different kinds of nerve cells. We have actual neurons, and then we have glial cells, lots and lots of super important glial cells that help to support, feed, move those neurons around so that they can do their jobs. Of course, that differentiation is all based on the activation of some genes and the deactivation of other ones. Once we have those genes turning on, and other genes turning off, so that our cells are growing the pieces and parts of nerve cells, those axons. We're going to start to move them around. So immature neurons that are not quite all the way done differentiating yet, are going to ooze around. And so this super amazing micrograph picture is showing us there's a dye that we can add to proteins on cells called GFP, green fluorescent protein. And it just makes literally the proteins and the cells glow in the dark so that we can watch things move. And you can see the nucleus here of the cell is literally oozing up and down and back up. Lots of chemical signals are helping to move the cell around in two different ways that we can move the neurons. So we can have those glial cells, those support cells, and this is the glial cell, and then here's the neuron. And so that neuron can literally just wrap its axons around the axon of the glial cell and slide on down to where it needs to be. We can also just move the cell body, and that's pretty much what's happening here, where the nucleus, the cell body was here, and then it's just kind of moving along the axon to kind of change which side of the cell is the axon side and which side is the dendrite and soma cell body side. We can get these axons to grow with lots and lots of chemical stimuli, which is going to affect not just the length of the axon, but also what direction that it grows in, and also how many dendrites start to grow. So we can add some chemical signals. Some of them are attractants. So I can have this chemical signal that's going to cause the axon to want to be toward it. We can also have chemical signals that are repellents that are going to push the axon away from it. And the collection of the attractants and the repellents are going to signal the axon to grow so that it gets to the target cell where we want it to be. Those chemical signals are also going to tell dendrites to grow or not grow, how many and in which direction those dendrites need to go. So lots of chemical stimuli in addition to the genes that we are activating and deactivating. So how even do those axons grow? We know that the chemical stimuli get them to grow in a particular direction, but how do they even grow? So we have what are called filopodia. They're like tiny little pod is foot. So there's these tiny little feet that stick out. And we have actin filaments. Remember that actin is one of our fibrous proteins. It helps to build our cytoskeleton, and so we're going to have these actin filaments that are going to push out the cell membrane, and then we can get more of our microtubules to kind of grow in and take places, and then we push these guys out more, again because of those chemical signals, and then we can grow the bigger microtubules along that way. So these filopodia, they can get crazy, crazy long. We have nerve cells that go from the base of our spinal cord all the way to the ends of our big toes. Single nerve cells with crazy, crazy long axons that go from our tailbones to our toes. Cool thing about axons, they can grow pretty fast, 5 millimeters a day. That's half a centimeter. It's like maybe half the width of one of your fingers is how fast these guys can grow because the actin is pushing out on those filopodia and getting these cells to lengthen, which will allow the microtubules to come in and then like set things up for Aurelios so that the actin can push a little bit more. All right. Let's see here. So in order to put together our synapses, remember that synapses are those connections between nerve cells. So from axon terminus to dendrite, synapses are the gaps between those nerve cells. So we're going to put lots and lots of special kinds of structures in those places to get these synapses to develop and to stick. This is a postsynaptic neuron. This is basically the end of a dendrite, cell body and dendrite. And you can see all of these little yellow guys are all of the axon terminals that are trying to form synapses with this nerve cell. So it's crazy cool how many connections can be made amongst all of our nerve cells. How that happens, so remember we talked about that actin. Those actin filaments are going to push these guys down. We're going to have some acetylcholine receptors. Acetylcholine, one of those neurotransmitters that we talked about when it came to pesticides, if you remember that, and the bugs that got paralyzed. So we're going to have some acetylcholine receptors here. We're going to have growth cones, so they're going to start to widen here a little bit. So instead of lots of little tiny fingers, we're going to have some fatter fingers that are merging, and this is going to become our synaptic motor end plate, or the axon terminus. And then we're going to start to build some neurotransmitters that are wrapped up in vesicles, being put there by rough ER ribosomes and Golgi apparatus. And then we're going to build those vesicles that can release those neurotransmitters across the synapse. So again, it's all about some receptors. It's about some other chemical messengers that help to stimulate this growth and stabilization, which then will lead to the formation of neurotransmitters in vesicles. We have lots of different kinds of synapses within the central nervous system. Central nervous system is brain and spinal cord. We can have axon and dendrite. We can have axon and the soma of a cell. We can have axon to axon. So this axon is stuck on that axon. And then we can also have dendrite. Most of the time I show you axon terminus connected to dendrite, or axon terminus connected to muscle fiber cell, but clearly that's an oversimplification. We have lots and lots of different ways to connect our nerve cells, lots of different paths to send messages along. So what's kind of amazing, so 36 weeks gestation, this is about a month prior to birth. This is newborn, this is 3 months, this is 6 months, this is 2 years. So you can see how much, how many synapses, how much growth between nerve cells is happening in these first 2 years of life. And so much nerve cell growth, so many nerve cell connections being formed. But here's the crazy thing, which is both bad and good. So when a synapse is being used, when we are teaching our kiddos how to count, how to speak, how to hold a crayon, chemical markers are going to be released. Those neurotransmitters are going to be released, going to strengthen and stabilize the synapse. And so those axons, those dendrites, are going to form the super strong connection that's going to stick. But if there aren't any chemical markers being transferred from one cell to the other, there's no need to keep that synapse, and so it's going to be pruned. So just like if you've got a tree that's growing crazy out of control, too many branches, more branches than is healthy, we're going to cut some of those branches off. That's pruning. We do that also with our nerve cells. And so you can see from 2 years to 4 years, 4 years to 6 years, there are actually fewer synapses, which isn't necessarily a bad thing. It looks like it's bad. We're like, oh no, more nerve cells is better. But not necessarily, because we want efficient paths. And if everything is helter-skelter all over the place, that inefficiency could lead to some developmental delays. And there is some thought that autism, one of the causes of autism might be that there's a lack of pruning, that going from here to here, kind of cleaning up the synapses that we don't need, might not be happening in people who have autism. And so that those extra synapses might lead to some of the symptoms of people with autism. So neurons or parts of neurons that we don't use, they're going to be destroyed by apoptosis. Apoptosis is when it's pre-programmed cell death. It happens on purpose. It's not because of an injury or a mutation. It is simply, I don't need this cell anymore. I'm going to take it apart and recycle the pieces into other things. So apoptosis, pre-programmed cell death. Neural pruning, we're going to take away those nerve cells that we don't need. So newborn babies, 11.2 million neurons in their thalamus. We think this is where a lot of our long-term memory is stuck. Adults only have 6.4. So we definitely have a reduction in the number of nerve cells in the thalamus. And I alluded to this a little bit earlier. We think that sometimes autism might be caused by or linked to a lack of neural pruning. So if we aren't taking away the cells that are not needed, that overabundance of nerve cells might be one of the causes, might be one of the many causes of autism. All right, so neuroplasticity. Plastic means that it's moldable, it is changeable, it is not static. It's plastic. And so the nervous system is actually always changing in response to the environment. Childhood, we definitely have greater plasticity. You guys learn so much more quickly than I do now because I've got old lady brain and I can still learn things, but it's way harder. My brain is just not as plastic as yours. So one of the ways that we can study neuroplasticity is in response to stroke. So stroke is when part of the brain dies because of either hemorrhage, bleeding is happening in the brain. So here, some of the blood vessels in the brain actually were broken and blood oozed out into the brain, put a bunch of pressure on the brain cells. This is a hemorrhagic stroke. And then ischemic stroke, there was some kind of blockage in the blood vessels. And so the blood couldn't get to where it needed to go, and all of this tissue, all that brain tissue is now dead because it's not being fed by blood. What's kind of cool is even in older people, if we lose one part of the brain because of a stroke, oh, that's a really bad color. If we lose one part of the brain because of a stroke, this part dies. Over time, it takes a while, it takes a lot of work, but over time, different parts of the brain can actually take over the functions that were lost when this chunk of the brain died. So super amazing neuroplasticity. Speaking of strokes, it's always good to know signs of stroke just in case someone in your family or whomever has a stroke. So FAST is the mnemonic to remember. So if the face looks uneven, if one of the arms is paralyzed, if the speech is slurred, then it is time to call 911. So for a stroke, signs of stroke, remember FAST. And so just a quick check of our objectives. Hopefully you're like, I totally remember all those neuron pieces and parts. I remember resting potential, depolarization, repolarization, hyperpolarization. I know a little bit more now about embryo structure. So zygote, morula, blastula, gastrula. We talked about the development, lots and lots of mitosis, lots of genes getting turned on and off. Neurulation is the formation of nerve cells when we differentiate from stem cells to nerve cells, and that's going to lead to brain, spinal cord, and then of course vertebrae. And if the vertebrae are not developing properly, spina bifida is a potential spina bifida is a potential disorder that could result if the vertebrae are not growing appropriately. And then we talked about growing neurons and pruning neurons and the amazing plasticity of your brains. Good work. See you next time.